Welcome students, myself Mr.P.P.Sutar, Associate Professor, Department of Chemistry, Gopal Krishna Gokhale College, Kolhapur. Students, in the previous lectures, we have seen about the UV visible spectroscopy. Today we are proceeding further. Now, today's first point is color and visible spectrum. Now, in the introduction topic, we have seen where is the visible spectrum situated in the electromagnetic spectrum. It is in between the range 400 to 800 nanometer. Why it is called as visible? Because it is visible to our human eye, and therefore it is called as a visible spectrum, or also called as a light. Okay. Now this light consists of 7 colors. Those are V, I, B, G, Y, O, R. Okay. Now see how the compound appears colored. Now when the visible light is fallen on the compound, then there are 3 cases. The first one is when all light fallen on the compound is totally reflected, then that compound appears white, because white light is fallen on the compound and it is totally reflected to our eye, and therefore the compound appears white. The second variety is when all light which is fallen on the compound is totally absorbed, and therefore there is no reflection of the light, and therefore our eye can't see any light, and therefore the compound appears black. Now in the third case, we can see the color to the substance. Okay. Now when the white light is fallen on the substance, then except some light, the remaining light is absorbed, and the reflected light is the color of that substance. It is clear from the next diagram. Okay. The first diagram I have cleared. When all light is absorbed by the substance, there is no reflection of the light, and therefore this compound appears black. In the second case, B, all the light which is fallen on the substance is reflected to our eye, and therefore the substance appears white. Now in the case C, when the white light is fallen on the substance except blue, remaining light is absorbed and blue light is reflected to our eye, and therefore the substance appears blue. Okay. And thus, if instead of blue, if there is a yellow color is reflected and all other light is absorbed, then the compound appears yellow. Thus there is a color to the substance. Okay, that is the color to the substance is the light which is reflected towards our eye. Okay. Now sometimes complementary colors are also seen. Let us see what are the complementary colors. In the table, the complementary colors are given. For wavelength 400 to 435 nanometer, when it is absorbed, then the violet color is absorbed, while the visualized color is yellow green. This is a complementary color. Okay. When the absorbed wavelength is in between 434 to 480 nanometer, the absorbed color is blue, but the visualized color is yellow. Thus there is a range of wavelength absorbed, the color absorbed, and the actually visualized color. Okay. And therefore there is a definite relation between the color and the constitution of the absorbing substance. That is, the relationship is very important because this decides which color will be having that substance. Okay. Now look the case of benzene. We are going to study the case of benzene. Now benzene, it is of course colorless. The energy requirement for the transition in benzene is quite high and cannot be met with the visible light. And so benzene reflects the entire visible light incident on it towards eye, and hence benzene is colorless. When benzene is converted into nitrobenzene, that is when the chromophore nitro group is added to the benzene, it is converted into nitrobenzene. Okay. And this nitrobenzene facilitates the delocalization. And this delocalization lowers the transition energy considerably and pushes the absorption to visible light range. Okay. So nitrobenzene absorbs in the visible region and it is pale yellow colored. And that is when chromophore is added, it favors the delocalization and therefore the absorption in the visible region. Okay. And therefore it can be said that highly conjugated organic compounds are colored as they have lambda max value in the visible region. Let us summarize. The color of a substance is due to selective absorption in visible range of radiation. Complement of the light absorbed get transmitted, so the color of the substance is the color of the reflected or transmitted light. This is complementary to the absorbed light color. More the absorption, darker the color, that is deepening of the color takes place. The color of an organic compound is due to the presence of chromophoric groups, that is the chromophoric groups are responsible for the color of the compound. Okay. Now let us go further for applications. Now these are the applications of UV visual spectroscopy. Now the applications of UV visual spectroscopy for structural diagnosis, that is complete diagnosis of the structure, is not possible because the information obtained is not sufficient about the knowledge of molecular structure. Now what information is got? Let us see one by one. Number one is determination of extended conjugation. We have seen when extended conjugation is present, there is an increase in the wavelength to the longer side, that is the lambda max is increased. Let us see, higher the lambda max value, which indicates longer the conjugation, and when lower the lambda max value, the shorter the conjugation, that is vice versa. Okay. And therefore the distinction between a conjugated diene and non-conjugated dienes structural analogous can be made easily. Now let us see. The first one is non-conjugated, that is the double bonds are not in conjugation, they are isolated, and therefore this type of conjugation is called as isolated or the non-conjugated, and therefore the lambda max value is 175 nanometer. When there is a conjugation, double bond, single bond, double bond, the lambda max value is 217 nanometer. Now here only conjugation is present. In the last case, there is a extended conjugation, double bond, single bond, double bond, single bond, double bond. That is, conjugation is increased, that is extended, and hence the lambda max value is also increased. It is 258. Thus, extent of conjugation can be determined by this method. Okay. The second is a detection of chromophores. A strong absorption usually is the characteristic of pi to pi star transition. If absorption occurs above 200 nanometer, a conjugated system of multiple bond is indicated. While weak absorption suggests N to pi star transition of isolated carbonyl group. And if this absorption is found in the region of 270 to 350 nanometer, an aldehyde or ketone is probably present. Okay. Now, somewhat stronger absorption between 200 to 260 nanometer may correspond to N to sigma star transition. Detection of geometrical isomers. Now, we know the geometrical isomers, they differ in the spatial arrangement of atoms. Okay, where here the example is given of stilbene. The first one is trans stilbene. The hydrogens are opposite. Aryl rings are also opposite. Cis stilbene, same side of the double bond. What is the difference? The lambda max value of trans isomer is higher than the cis isomer. Let us see. Lambda max value for trans stilbene is 294 nanometer, while for cis stilbene it is 278 nanometer. And also the Emax value is also more for trans stilbene, while it is less for cis stilbene. Let us see another example. Cinnamic acid. The first variety is trans, second is cis. When there is a trans variety present, the lambda max value is 272 nanometer, while the cis cinnamic acid have the lambda max value 268 nanometer. Also, the Emax value for trans is more than the cis isomer. Now, why this is happen? So, the close proximity of two substituent groups in cis isomer hinders or stops coplanarity of pi system, lowering the resonance to some extent, and this increases the delta E value, making the transition more energetic. So the absorption shifts to shorter wavelength. Okay. Then fourth one, the structure of organic compounds. UV spectroscopy is one of the methods used for structure elucidation of organic molecules. However, its utility is confined to know the presence or absence of chromophore. From the location of peaks and combination of peaks, it can be concluded that whether the compound is saturated or unsaturated, whether heteroatoms are present or absent, and this the structure of organic compound can be identified. The application next is quantitative analysis. Now, UV spectroscopy is for the quantitative determination of compounds that absorb UV radiation, and this determination is based on Beer's law, as we have discussed in earlier lectures. The relation is A, that is absorption, is equal to epsilon into B into C, where epsilon is the extension coefficient, C is the concentration, and B is the length of the cell used. Qualitative analysis. In the qualitative analysis, UV spectroscopy is specially used for characterizing aromatic compounds and aromatic olefins. Identification is done by comparing the absorption spectrum of the spectra of known compounds, and from which we can have the knowledge of quality, that which elements are present. Then the strength of hydrogen bond also determine. The strength of hydrogen bond can be calculated by taking UV spectrum in polar and non-polar solvent. When, for example, acetone, when polar solvent like water is used, there is a decrease in the wavelength, that is a 264 nanometer. While when it is done in the non-polar solvent like normal hexane, the wavelength is 297. The difference between these lambda max values in the polar solvent and non-polar solvent directly gives the strength of hydrogen bond. The difference, okay, why? The difference is calculated, whether the bond is strong or the bond is weak. Okay. The last is detection of impurities. Now it is one of the best method for detection of organic impurities present in the sample. A comparison of spectra of sample, which is whether it contains impurities or not, is done with the standard material at specific wavelength. Presence of impurity is hinted by the appearance of additional peaks in the spectra of the sample, and this can be easily noticed. That is, the standard gives regular appearance peaks, while the sample will give, if it contains impurity, additional peaks will be obtained, and thus we can identify whether the sample contains impurity. Okay, students, with this point we have completed all points from the topic UV visible spectroscopy. Now, students, you have to go through all the topics, see the important points, that is the definition of chromophore, exochromes, then the shifts, that is a bathochromic shift, hypsochromic shift, and then the effects, that is a hyperchromic effect, hypochromic effect, and the problems solving using Woodward-Fisher rules. The problems on dienes and enones are asked in the examination, and therefore try to solve more problems on dienes and enones. Thank you.